Hi folks, welcome to this video for National 5 biology students where we're going to look at the apparatus and techniques section of the course, which are a list of apparatus and techniques that you need to know about for the exam. And we're also going to look at control, reliability and validity, as well as factors in experimental design and talking through how you would answer questions in those, because again, that's an area where students commonly lose marks within the skills section. So we're going to kick off by looking at the apparatus that you need to know about. A lot of this will be familiar to you, so it won't be something we'll have to go over in any real depth, but we can see we've got a list here. So we've got a beaker. Okay, this is a beaker in the top left there. A balance, which we use to measure the mass of various things. We've got a measuring cylinder, which we use there. It's a tall cylindrical tube that's used to measure the volume of various liquids and solutions. We've got a dropper or pipette, which is used for dispensing small quantities of liquid. We've got test tubes and boiling tubes, which look a little bit like these here, which are the vessels in which we put chemicals to react and mix together. We've got thermometers. Thermometers are used to measure the temperature within a solution, within an experiment. Funnels, which we use to separate materials. We can put filter paper in there and we can separate solids from liquids, etc. We've got a syringe as well, which is used for accurate dispensing of solutions. We've got a timer or stopwatch over here, which, as the name suggests, is for recording time. We've got a microscope here, which, again, you're probably all familiar with, which is used to magnify very small things that we want to look at in biology. The Petri dish, which we use for growing microbes. We usually put a wee bit of agar or whatever onto the Petri dish, and we can grow cells and microorganisms and other things on there. A quadrat, which again, this is part of the course, so you really should have covered quadrats, but just in case, quadrat's this square measuring instrument and it measures the abundance of usually plants within an area. Can also be used for very slow moving organisms, but yeah, usually for the abundance of plant species in an area. A pitfall trap, which is used for trapping and therefore sampling the number of invertebrate organisms, ground living invertebrate organisms within an area. There's the light and moisture meter, which you can see in the bottom right here. This one's a light pH and moisture meter, so it's got a 3 in 1 there. And what that allows you to do is to measure abiotic factors within an environment, which is going to be useful in terms of determining what might affect the abundance of an organism. And lastly, we've got a water bath here. Okay, this is a big water bath. Not very efficient water baths. They basically heat up water to a particular temperature and keep it there. It's useful if you're keeping, I guess, stuff at a constant temperature for a long period of time. It means you can control temperature in in like a solution that's in a test tube or something, because you can immerse the test tube into that water. These days, there's more modern devices that can maintain temperature, like thermotubes, for example. So water baths are a slightly older and less efficient way to do that, but that is the one that the SQA want you to know about for your exam. So a water bath is there for maintaining the temperature of solutions. Okay, it's good if you've got a question that asks about how would you modify this to ensure that the temperature is kept the same throughout. You would say use a water bath. Okay, the techniques that you need to know about for the exam, these are the ones that could come up in your exam and you could be asked about. First one's measuring enzyme activity. So that's usually a set of experiments that you can do, various enzymes, various substrates, and you'd measure the activity in a whole number of ways. It might be the gas that's released, and you can use detergent in there to measure the height of the froth that's made, or you can use a gas syringe to capture all the gas that's released from the reaction, and the more it moves the syringe, the more gas is released, and so on. There's a number of different ways that you can measure enzyme activity, but the experiments involving various enzymes and substrates will be able to be measured, okay? And you've probably done a few of those in class at some point. Using a respirometer, we've got a respirometer here top right. So a respirometer is where you would place an organism, a living organism, a respiring organism, into one of these little chambers here, and we would measure the movement of a liquid through the respirometer there, and that would indicate the level of respiration that occurs due to the gas that's made by the organism. Okay, the carbon dioxide that's made sometimes a respirometer might look a bit different, and you've got the capillary tube going into some lime water or something to measure the carbon dioxide secreted by the organism. But this is a respirometer, okay? And as I say, they might look just slightly different from this one, but being familiar with them is useful for the exam in case they ask about it. Measuring transpiration using a potometer. So we've got one bottom right here, a potometer, where we pop our plant into the potometer, and again we see the movement of the coloured liquid, or just a clear liquid if you can read it okay in the potometer, as it passes up this scale. So basically, as more water is drawn up by the plant, the scale, the bubble, the air bubble that you've trapped in here, will move along the scale, and you can measure the rate of transpiration in the plant. Measuring abiotic factors, we talked about it in the last slide, but we'd use something like a soil or pH meter, a light meter, thermometer. These kind of instruments that we talked about in the last slide, they would all measure our abiotic or non-living factors. Measuring the distribution of species. So again, the quadrat and the pitfall trap that we looked at in the last slide are both examples of how we could measure the distribution of a species. Using a transect line. So a transect line is where, you can see in this middle diagram here, where you would form a line. Quite often use a big long bit of string, but you don't necessarily need to do that. And you're going to be measuring the difference in abundance of an organism between two different conditions, two slowly changing conditions. Okay, in this case, they're looking at altitude, so they're taking samples all the way along this line here, and at each area where they're taking a sample, the altitude is getting smaller. Okay, but the more common than that, maybe something you've done in your class, is you might look at a shaded area and an open area, and you make a transect line between the under the trees where it's shaded all the way out into an open field, and you would then take a series of measurements along that line from one differing environment to another. And then lastly, measuring the rate of photosynthesis. So you can do that in a number of ways. You can use like a water plant and measure the bubbles of gas that are produced by it, the oxygen gas that's produced by the plant. That's a nice easy way to do it. But there's a few different ways of measuring photosynthesis within organisms as well, and you'll have done a different one in your class depending on what your teacher has decided to do with you. But you need to know how we do that, and as I say, measuring bubbles of oxygen gas that are produced is one way that they do it. So there are three main key terms that you need to know about in terms of experimental design for your exam, and the SQA say that candidates should be familiar with the terms control, validity, and reliability. And you should be able to comment on these in experimental setup questions. So that's what we're going to continue to do through the rest of this video, is to look at those three and to look at some of the questions that might be asked of you in the exam. So first of all, control. Now, a control is set up to compare results of an experiment when no treatment is applied. So, for example, if you're looking at the effect of exercise on heart rate, the no treatment, the control that you would have for that experiment is to take away the exercise, and you would measure the heart rate at rest without any exercise. Okay, so that's a control, and we'll talk more about that in a moment. Validity, so other variables are controlled to ensure that any effect is likely due to the variable being changed. Okay, so we change one variable and we measure one variable, and every other variable has to be kept constant and controlled throughout the whole experiment. So again, we'll talk more about validity in a few moments, and how we would apply that to questions in the exam. And reliability, where consistent values and repeats of an experiment are obtained. Now, whenever I'm teaching my classes, I always remind them of these little phrases that will help them remember what each of these three key terms actually is referring to. So we've got COCO, so control, compare. Okay, control, compare. If you can remember that, that will sort of move you in the right direction in terms of remembering what a control is. Okay, it's a comparison from when we have the treatment to when we don't have the treatment. Okay, depending on what that treatment is. VAVA, validity variables. Okay, so when a question is asking you about the validity of an experiment, it's referring to the variables within it. Have the variables been controlled? Is there only one variable being altered during the experiment, etc., etc.? RERE is reliability repeat. And again, if you're asked about how you would improve the reliability of an experiment, generally speaking, you're saying something like, we are repeating the experiment, okay, or being more specific, we are repeating the experiment at all concentrations, or whatever it happens to be, okay, whatever the specific question is. So variables and controls, Let's talk about them sort of together, first of all, because a control experiment is essentially where you're changing one feature from the main experiment, where you're removing the thing that you're investigating, but everything else must be kept the same. So controls and validity kind of go hand in hand when we're talking about them. So when you do an experiment, you should only change one feature of the design, and that's the variable that you're investigating. Okay, so that might be the intensity of the exercise is going to be the thing that might change in terms of measuring heart rate. You might be looking at the concentration of a substrate if you're looking at enzyme activity, or the concentration of the enzyme for that matter. You might be looking at changing temperature if you're looking at the rate of respiration in an organism as well. Okay, so you're only changing that one variable, everything else must be kept the same. All right, and an example of a control experiment would be to ask the subject to measure their heart rate in the absence of the variable you're investigating. In this example that I gave earlier on, that is meaning doing no exercise, okay? So it's the absence of the variable that you're investigating. So let's have a look at some examples. Sometimes we have to replace the variable that we're investigating with an alternative rather than nothing, like the example below. So what would you do to make this into a control experiment? Okay, so what alteration would you make to make a control experiment for this main experiment here? And you can pause the video and work this out yourself. But it's just some background here. It says a student sets up an experiment to investigate the breakdown of starch by the enzyme amylase. So the purpose of this experiment is to investigate the breakdown of starch by the enzyme amylase. So we're investigating the enzyme amylase here, okay, and its ability to break down starch. So therefore, if we were wanting to investigate amylase's ability to break down the starch, then that would be the thing that we would replace or remove from the experiment. Now, normally, if we want the absence of the factor we're investigating, so the absence of amylase, and that's fine. But the problem with this experiment is if we just take the amylase away, we're not just changing that one thing in the control experiment, we're also taking away 1 milliliter of solution. So in that original experiment, we've got 10 starch and we've got 1 amylase, okay? So that's a total of 11 milliliters of solution. Now, if we take away the amylase for our control experiment, we're left with 10 milliliters of starch solution. So the volume has also changed in addition to us removing the amylase. So what we've got to do is make sure the volume's the same. So what could we put in there that would replace amylase and not have an impact in the experiment, but still give us the same volume? Something like water would be a great replacement. Okay, so we'd replace that 1 mil of amylase solution with 1 mil of water, and then that would form a control experiment. Having a look at this one, we've got an investigation that was carried out into the effect of pH on the activity of the enzyme pepsin. Okay, the effect of pH on the enzyme pepsin. And I'm just going to underline the important things here. Okay, pH, enzyme pepsin. So what would be the control experiment for this one? Well, they're changing the pH to see how the enzyme is going to react at those various pHs. So a control experiment would be the absence of the thing we're investigating. That's the enzyme pepsin. So it would be exactly the same setup. Okay, still got these 6 little wells here. Each of them should have the different pHs and so on. But instead of adding the enzyme pepsin, we would add either nothing or the equivalent volume of water. Okay, so whatever they have added in terms of volume for the enzyme, then we'd add the same in terms of water. For this one, we've got the rate of transpiration in plants been measured using the apparatus below. Okay, as the plant transpires, the coloured water is drawn up the glass tube and its volume is measured over a set period of time to give the rate of transpiration. Okay, so how would we set up a control experiment for this? Well, the thing that we're investigating is this plant, right? The rate of transpiration in plants. So we're measuring the rate of transpiration, and the plant is the thing that's going to be transpiring. So what we have to do for a control experiment is to remove this plant. Okay, now we could just remove the plant and not have it there. That would be a suitable control. But what would maybe be even better, and where we're changing even fewer things, is for us to do something with the plant so that it still has the same mass, it still is there, but it's not going to carry out transpiration. So, like most processes inside our cells, in plants, animals, etc., they are controlled by enzymes. And so, if we were to boil this plant, then we would denature all those enzymes, and that would mean the reactions wouldn't happen. Okay, so maybe like a boiled version of this, okay, or a dead version of this would be a suitable replacement as well as just removing the leaf altogether. For this next one, we've got an investigation carried out to find the effect of pH on fermentation in yeast. Okay, so we're changing the pH, we're measuring fermentation that happens in the yeast. Okay, so what would we change here to make this a control experiment? Well, what could we remove that would remove the thing that we're investigating? We're investigating fermentation in yeast, so therefore removing the yeast would be a suitable control. Okay, so everything else the same, the exact same setup, but minus the yeast. Next one, a student is investigating the effect of temperature on the rate of respiration in germinating peas. Okay, so this is the thing we're changing, the temperature. The rate of respiration is the thing we're measuring, and the germinating growing peas are the organism in which we are carrying out the experiment. Okay, and you can see the setup here. Now, again, if we want to set up a control for this, we would be wanting to remove the thing that we're investigating, which is the rate of respiration in germinating peas. So we can remove the peas from the experiment, and that would be a control experiment. But again, what would be even better than just removing the peas? Well, keeping the peas in there, but making sure they can't germinate. Okay, and how would we do that? We would boil them. Okay, boil them denatures all the enzymes, and therefore it can't carry out respiration, which, as we know, is a enzyme controlled reaction. Okay, so we're going to remove these peas and we're going to replace them with some glass beads or something similar. Okay, but everything else in the experiment, every other part of it, would be exactly the same, minus the removal of the germinating peas for something inanimate like a glass bead, or to actually boil the peas and put boiled peas in there instead. That would also do a good job as a control experiment. The next one, an experiment set up as shown to measure the transpiration rate of a plant at room temperature. The mass was recorded at the start, and again after 6 hours. The results are shown in the table. So if we wanted to compare this experiment to the absence of the thing that we're investigating, we would simply remove the plant, okay, or put in a boiled version of the plant, or a plant that's been heat treated so that everything has been denatured inside it. Okay, so something like that would be a suitable control for this one. And here's an example of a question from an exam paper which came up very recently, actually, in the last 2 years, I think, either last year or the year before. This one says, a respirometer was set up to investigate respiration in maggots. So that's fine. We're investigating respiration in maggots. Now, we did this one earlier on where we looked at the germinating peas, okay, where it was in a respirometer. And this question is asking us which of the following would be a suitable control for the investigation. Now, again, remember, we are looking to compare it with the absence of the thing we're investigating. Now, we're investigating respiration in maggots, so that's the thing that will have to be absent within our control, okay? Now, because A has maggots, we know it's not going to be that. B has glass beads in place of the maggots, and seems to have everything else the same, so we'll come back to that in a moment. C has the glass beads, however, it's missing this chemical here that's present in the original experiment, so therefore they've changed the glass beads and they've taken out the chemical, so they've changed two things, so that can't be a control. And lastly here, again, we've got the maggots, okay, we should have really removed those maggots, but we've also removed our chemical down here, which we shouldn't have done either, okay, so we can eliminate D as an option. So the best option here is going to be B. Okay. Now, in terms of validity, results are only valid if you only change the variable you are investigating. Okay. You only change the variable you're investigating. If you change more than one variable, the experiment becomes invalid. Okay. Remember that word, invalid, if you change more than one variable. Now, the variable you can change is called the independent variable. Now, technically, you don't really need to know that for National 5. It's unlikely they'll ask that, or if they do, they'll explain what it is. But the independent variable is the one you can change. That's the thing that you are altering in terms of the conditions within your experiment. Okay, so it might be the temperature, it might be the concentration of something, or similar to that. Now, the thing that you measure, okay, i.e. your results, are called the dependent variable, okay, because they depend on what actually happens in the experiment. Now, an example of this, if we look at the next slide, we need to try and find an issue with the 4 experiments being used. Okay, so this diagram shows 4 experiments carried out as part of an investigation into the rate of respiration in yeast. Okay, respiration in yeast. Now, which of these 2 could we validly compare? Okay, we're only allowed to change one thing when we're doing a comparison. Okay, there's only one thing that can change. Now, this is looking at the rate of respiration in yeast. So what one thing could we change that would show a difference here? Well, let's have a look at the 4 experiments. Experiment 1 has 1 gram of yeast. It has 2% glucose solution and is a water bath set up at 22 degrees. Experiment 2 also is 1 gram of yeast, but this one has 5% glucose solution and again is set up at 22 degrees. So if we have a look here, 1 and 2, experiment 1 and 2 only have one thing that's been changed, and that's the percentage of glucose solution. Okay. Experiment 3 has got 1 gram of yeast, 5% glucose, and the water bath is set up at 30 degrees, okay? Now, again, if we look at experiment 2 and experiment 3, we can see there's only been one thing changed. The yeast is the same, the glucose solution is the same, but the temperature has been altered, okay? And then lastly, we've got 2 grams of yeast, 2% glucose, and the water bath is set up at 30. Now, we can't compare 3 and 4 because we've got the grams of yeast been different and the percentage of glucose solution been different. So we've changed 2 things here, okay? So 3 and 4 cannot go together. 3 and 2 could go together because we're only changing one thing, and that's the temperature. 1 and 2 can go together because we're only changing one thing, that's the percentage of glucose solution. But 1 and 3, for example, couldn't go together because we're changing 2 things, the percentage of glucose and the temperature. And 1 and 4 couldn't go together because we're changing 2 things again. The grams of yeast would be different there, as well as the temperature. Okay. So which of those could be validly compared? Well, we can compare 1 and 2 together, or we could compare 2 and 3 together. Okay. All the other ones could not be compared to each other because we're changing more than one thing when we're doing that. Let's have a look at this one. For a valid conclusion to be drawn, which 2 tubes should be compared to show the effect of temperature on germination? So the effect of temperature. Now, if you're looking at the effect of temperature, that is the one thing you're allowed to change, but everything else must be kept the same. So if you want to pause the video just now, you can see which of the 2 tubes would you be able to compare only changing the temperature, okay? Comparing them, looking at the effect of temperature. And hopefully you've worked it out. We're looking at this one here, which is 5 degrees, has dry cotton wool and 20 seeds. And this one here, which also has 20 seeds, also is dry cotton wool, but does change the temperature, okay? So tubes 3 and 4 there would be valid in terms of comparing them in terms of temperature, okay? Also, if we had a look at tubes number 2 and 5 here, we can see 2's got 10 seeds, so does 5. It's got wet cotton wool, so does 5. But the only thing that differs is the temperature there, okay? Everything else remains the same, okay? I hope that makes sense and we're all following this so far. The next one, an investigation was carried out to compare the rate of oxygen gas produced by 2 species, S and T. Okay, which diagram would show the setup for species T that would allow a valid comparison in the rate of oxygen between the 2 species? Okay, so we're only allowed to change one thing. And it says here that the thing they're going to change is the species. Okay, so species S will be replaced for species T. But every other element of the experimental design should be exactly the same as this original experiment. The only change should be that species T replaces species S. So let's have a look at the various options here. Now, A, does that look very similar? To me, it does. A looks very similar to the one above, with the only real difference being that we've got species T in place of species S. We'll come back to A in a minute. Let's look at B. We've got species T again, but this time if we look at the length of that plant, okay, that is much longer than the length of the original plant. So therefore, they've changed not just the species, but also the length of the plant there. So that would not be valid, okay? We can get rid of B as an option. If we look at C, now it's got species T there, which is great. The length of the plant is also the same, which is great. Everything else looks perfect, apart from the lamp is further away in this experiment than it is in the original one. So that means we've changed both the species and the distance that this lamp is. So that's 2 things we've changed, not a valid comparison. For D, again, we've got species T, which is fine, but the length of this plant is far too long. Okay, so that's a second thing we've changed. And actually, this lamp is too far away as well. So they've changed 1, 2, and 3 things compared to this original experiment. So again, that is very much not a valid comparison. So our answer is going to be A for this one. Okay, A is the one that will show a valid comparison. have a look at this one. This was from, I think, last year's paper. Which two tubes should be compared to show the effect of light on photosynthesis? Okay, so they're investigating the conditions needed for photosynthesis. So which two should be compared to show the effect of light on photosynthesis? So if you're looking at the effect of light, the two that should be compared are one that's in the light and one that's in the dark. Okay, that's the two conditions that you should be comparing. Every other element has to be kept the same. Okay, so let's have a look at the options here. Now, 1 and 2 are both in the light, so we can't compare them. Okay, 1 and 3, well, that's light and dark, so we can compare them. Okay, in terms of the condition. If we also look at the carbon dioxide concentration, that is also the same. Okay, the only difference being the light and the dark. So 1 and 3 is a possibility. However, if we look at the answers, 1 and 3 is not an option there. Okay, so we need to find another one there. What about 1 and 4? Well, 1's in light, 4's in dark. That's perfect. That's what we want. But if we look at the carbon dioxide concentration, those are different between 1 and 4, which means we're not only changing the condition, light and dark, we're also changing a second factor, carbon dioxide concentration. So that would not be valid as a comparison. If we have a look at 2 and 3, that's not going to work, even though there's one light and one dark, because the concentrations are different. But if we look at 2 and 4, 2 is light, 4 is dark. That's what we want. 2 is 0.3% concentration. 4 is also 0.3%. So they've kept the concentration the same, but the only condition they've changed is whether it's in the light or the dark, which is what we want here. Okay, we want to look at the effect of light on photosynthesis. So our answer there is going to be D, 2 and 4. Okay, now validity, we talked about it earlier on. Validity is VAVA. Okay, so that means validity equals variables. Okay, so when you are asked in the exam something about the validity of an experiment, you should immediately be thinking, okay, what variables have been kept constant and controlled and which variables perhaps haven't? Because if those variables have not been kept constant and controlled, then the experiment would be invalid. Okay, in order to keep it valid, for good validity in the experiment, all the variables, apart from the one you're changing, must be kept constant. Okay, now the factors that could affect the result if they're not controlled, these variables as we call them, are things like the mass of tissue or organisms, the time the experiment's left for, the temperature of, for example, the beaker or test tube or the solution or something similar, the volume of any potential solutions, the concentration of any solutions, the pH of the solutions or the pH of the soil or the pH of whatever it is. If we're talking about a living organism, the species or type of organism could be a factor. The abiotic factors such as light intensity, soil moisture, pH, these would all affect the result of the experiment if we did not control them. So we have to. The number or abundance of organisms, the size of the test tubes or beakers could have an impact, and other units of measurement as well. Things like the diameter of something, the depths of something, the length, the width. All of these things will have an impact on the results if they are not controlled and kept constant throughout the experiment. Okay, so that's quite a big old list for you to remember. The ones that are probably the most common ones that come up in the exams are mass, time, temperature, volume, concentration, and pH from time to time as well. The other ones are quite specific to a type of question, okay? So they won't necessarily come up in every single one of these examples. But the way I always teach it to my classes, I always say, well, when I was a youngster back in the 80s, we used to watch a television channel called MTV, okay? So it was the MTV channel. And that is a way that I remember the variables in an experiment, because I think, okay, M for mass, T for time and temperature, V for volume, and C for concentration. And if I can remember those 5, then I've got a good shot of being able to list variables we have to keep constant within an experiment. Okay, so just remember Mr. White's childhood when he was watching MTV in the 80s and 90s. MTV channel will give you your variables that you need to know for your experiments in the exam. So let's have a look at how we can apply that. So it says the apparatus was set up to investigate the effect of temperature on fermentation in yeast. Okay, you can see these experiments become a little bit repetitive once you've looked at enough exam papers. Now, this investigation was carried out at 15 degrees and then repeated at 20 and 30 degrees, and the number of gas bubbles produced was counted. Okay, so gas bubbles was counted. Now, the thing I always like to do before I even get into the questions on these is I like to underline things that I think are important. So you'll notice that I've underlined what they're changing, the temperature. what they're measuring, fermentation in yeast, the various temperatures that they're changing, 15, 20, and 30 there, and then the way that they're measuring it, and that's via gas bubbles being produced, okay? And remember, gas bubbles produced as a indirect measurement of the effect of fermentation in yeast, okay? So it's an indirect measurement, and that is an important thing to remember. Sometimes when we can, like how else would you measure fermentation other than to count maybe the gas released by the organism or whatever? So it's an indirect measurement, it's not a direct measurement. And we'll talk more about that actually when we look at the scientific literacy questions in a different video. So which two variables, to keep us on track here, which two variables should be kept constant during the investigation? So let's have a look at what they've listed for us, because it's multiple choice here. We've got some options. So temperature, okay. Now, should temperature be kept constant during this investigation? Well, no, because it's actually the thing that we are changing. It's the one thing we are going to change, okay? So temperature is not going to be an option for us. And I noticed also that temperature is an option down for C there as well, which means we can eliminate both A and C as options straight away. Let's look at the other options. B, the type of yeast. Now, they don't mention the type of yeast, and we would certainly want to keep that the same. So that's probably a good one. And the time that it's left for. Well, again, that's probably going to be a good consideration, isn't it? How much time we leave the investigation for. If we look at D there, sorry, we've got the type of yeast again, which is fine. That's something we should consider. Now, the carbon dioxide concentration is an interesting one, because that is something that is probably an important consideration, but it's very hard to keep that constant during the investigation, because what's going to happen is the yeast is going to be continually producing carbon dioxide throughout the experiment. So it's not something we're going to be able to keep constant as a result of that. Okay, so that's going to fluctuate, which means it can't be D, and your answer there has got to be B. Okay, the next one, we've got an investigation into the distribution of woodlice in a woodland. Students collected and counted them every day for 5 days. Okay, that's important, using the sampling technique shown below. Okay, so we get the distribution of woodlice counted every day for 5 days. What's this sampling technique called? That's an easy one. That's a pitfall trap. Hopefully, we remembered that from our techniques earlier on. We use a pitfall trap to measure the abundance of a ground living invertebrate within a woodland area or something similar. But it's really part two that we're interested in for this question. So identify two variables that should be kept constant when setting up this sampling technique. Okay, so what kind of things do we want to keep constant? Now, this is a tricky one, because things like our mass and our temperature and concentrations and all that don't necessarily apply to this one. So what would we want to keep constant when setting up this sampling technique? Well, we talked about other measurements, things like the size of things and the depth of things and other factors, and they probably more come into play in a question like this. Okay, so something like the depth of our pitfall traps. Okay, the depth that they are is probably an important thing to keep constant. The width as well. Okay, or we could just say overall the size of the trap. Okay, that's something that we'd want to keep constant. Other things that we might want to keep constant are the type of leaf that we cover up the trap with as well, or the gap that we leave between the leaf and being able to fall into the trap. We'd want to keep that the same, or the depth to which we dig the trap into the ground to ensure it's level with the soil. That's also something we'd want to keep constant. Okay, so with a question like this, it's not quite as simple as some of the other ones, and we have to think about like a specific example for this circumstance. Okay, and hopefully I've given you a few ideas there to be able to answer a question like that. But anyway, the point is that we understand what has been asked in the question rather than us sort of memorising the answers, okay? And what has been asked here is, apart from what we're investigating, what should be kept constant when setting up this sampling technique, okay? And those are some of the variables. They are the things which could affect the result if they are not kept constant throughout. Okay, an experiment was set up to investigate the time taken for starch to be produced. All the previously made starch was removed from the potato extract. We'll get 2 rows of dimple tile, of the dimple tile set up as shown in the diagram. Okay, fine. So we've got this molecule here, G1P, and we've got some potato extract. We've got G1P and distilled water, okay? Now, the potato extract contains an enzyme, okay? It contains an enzyme. Okay, I'll just write that in there so we remember that. Contains the enzyme for this experiment. Okay. Now, why is that important? Because we are investigating the time taken for starch to be produced. Now, when G1P and the enzyme combine together, then the G1P will be synthesized into starch. Okay. Um, so what the experiment says here, at 5 minute intervals, we're going to be testing for starch. Okay, i.e., seeing how long it takes for this reaction to take place and for starch to be produced. We've also got a control experiment set up here. Okay, because we've got the G1P, that's the substrate, and we've also got some distilled water. That water is there in place of the thing we're investigating, which is the enzyme in the potato extract. So that's an example of a control experiment. So we would expect no result in that control experiment. Okay. And what that would prove is that it's actually the enzyme in the potato extract that's causing any change in result. But we're not really that interested in the experiment itself, okay, and the results of the experiment. What we're more interested for this question is to state two variables that should be controlled to make this experiment valid. Okay. So what kind of things do we need to control to make the experiment valid? Okay. Now we can think about our MTBC here for this one. So we can think about things like the volumes. Okay. Now there's no mention of any volumes there. So the volume of the potato extract or enzyme, the volume of distilled water, those would both be acceptable answers there. The concentration as well doesn't mention any concentrations. So the concentration of the enzyme, for example, is a factor. The concentration of the substrate, that's the G1P, would also have an impact on it. And we could also talk about um, the the pH as well, because we're talking about solutions there. Okay, so the pH within the potato extract solution as well. Okay, so a few different possibilities for us there. Now, lastly, we're going to talk about reliability. Where consistent values and repeats of experiments are obtained, we would say that we have a set of reliable results. Okay, so if the result of the experiment is 5 the first time you do it, it's 5 the second time you do it, it's 5 the third time you do it, we would be able to comfortably say those results are reliable. However, if the results were 1, then 10, then 2, then 9, we would probably say those results are not particularly reliable. And in order to improve the reliability, we would probably repeat that experiment several more times to try and get closer to the true values, where we get more consistent values. And essentially, when you've got lots of fluctuation in your data like that, the more repeats you do, the more likely you are to get closer to the true value. So the line that you guys need to know for your National 5 exam, okay, and it's not as straightforward as this in reality, but for the sake of the National 5 exam, when it asks you how you could increase the reliability of an experiment, it's usually asking you to say something like, to make an experiment more reliable, we would repeat the experiment, okay, or repeat the investigation, depending on what it says in the question. Okay, so that's what they're looking for most of the time for this type of question. Now, when we're thinking about reliability as well, we're thinking about something like sample size. So basically, if you're doing a human study, for example, the greater the sample size, the greater the number of humans involved in the study, the more reliable that becomes. Okay. If you think about, if you're investigating a drug, for example, and you only test that drug on 5 people, that's not representative of the whole population, the whole of humanity. So we would say that that sample size is too small for the results to be reliable. However, if we increase the sample size, we used more subjects, more humans, then that would increase the reliability of the results that we get. Okay. And it's the same with, you're doing experiments. You might be throwing a quadrat, or you might be measuring using a light meter. The more repeats you do, generally speaking, the greater the reliability of your results. Okay. So if an experiment is repeated once, you can increase reliability by repeating it 5 times. Or if your experiment involved 5 humans or 5 plants, you could increase the reliability by using more humans or plants in the investigation. Okay, so I hope we've got the gist of reliability. And I always say, remember reliability, repeat, and that will help you when you see this word in the exam. So let's have a look at this question again. Now, we talked about it earlier on in terms of the variables, but I'm going to jump down to this question at the bottom here, which asks us about making the results more reliable. So in this case, we are setting up a pitfall trap. How could we make the results of the investigation here more reliable? Well, simply, we would set up more traps. Okay, we'd set up more traps within that area. If we have 10 or 15 traps, then we're more likely to get a reliable representation of the number of woodlice in that woodland. So something as simple as that would get you the mark for that question. Here's another one for reliability. A group of scientists investigated the effect, this time of temperature, on the growth of genetically modified cells. That's fine. The GM cells were grown in a nutrient solution at 20 degrees. This was repeated at 30 and 40. Okay, so we're changing the temperature here, using fresh nutrient solutions at each temperature. Which of the following would improve the reliability of the results? So as soon as you see reliability, you should be thinking in your head, repeat, reliability, repeat. So would we set up the nutrient solutions containing GM cells at 25 and 35 degrees? Well, that would increase the range of temperatures that we use, but not necessarily increase the reliability of our results. Would we set up 10 nutrient solutions containing GM cells at each temperature? Well, that sounds good to me because that sounds like we're doing 10 repeats at each temperature, which sounds to me like it would be reliable. Do we use the same concentration of nutrient solution at each temperature? Well, that's true. That's good practice, but that would improve the validity, not the reliability. And using the same types of GM cell at each temperature, again, that would improve the validity, not the reliability. Okay, so the answer is going to be B for that one. Okay. Next one, 3 students carried out an investigation into the effect of exercise on heart rate. Each student measured their heart rate after completing the same exercises. After each student's heart rate was returned to its resting state, they repeated the investigation. Which of the following would improve the reliability of the results? Okay, so this involves humans, this experiment, so we have to think about it in just a slightly different way to other experiments. So let's have a look at the options. Increasing the rest period before repeating the exercise. I don't necessarily know if that's going to improve the reliability of the results. Okay, that's more to do with the factors that might affect the results, i.e., the validity. Completing the exercise in a different location. Again, I don't think that's a very good practice because that's, again, changing a factor. That's making it less valid, but not necessarily anything to do with the reliability. Changing the type of exercise each time. Again, doesn't sound like good practice, does it? Changing the type of exercise. We want to be keeping things the same throughout an experiment. So again, that would make that experiment invalid actually if we did that. And then lastly, increasing the number of students exercising. We're increasing the numbers, okay, increasing the number of participants. That will increase the reliability of the results, okay? So remember, if our humans involved or other animals involved, more people, more subjects, more animals, more plants involved would increase the reliability of the results. Okay, and then we've got some students investigating the distribution of some organisms on a rocky shore. That's fine, okay. Starting at the lowest tide level, there were quadrats placed every 2 metres along a single transect line, okay, and the number of barnacles and mussels were counted. Okay, perfect. So we've got 10 quadrats there on the table, right? And they've counted the number of barnacles and mussels at each quadrat as they've gone from the low to the high tide level. So our question asks us, describe how the reliability of the results could be improved. Okay, so again, think reliability, repeat. What could we do? We've used 10 quadrats here, okay? What could we do to increase the reliability? We could use or throw more quadrats, okay? So increase the number of quadrats that have been thrown in the experiment. Or we could simply say, repeat the whole investigation or experiment again, okay? Repeat the investigation again, repeat the experiment again. Something similar to that would get you the mark for this particular question. Okay, folks, so I hope that was helpful for you and that you understood reliability, validity control, and some of the practical techniques and apparatus as well a little bit better after that video. As always, just leave any comments that you've got about how useful it was, or if you've got any questions that you need to clarify in terms of the content there. If there's anything at all you want to ask me, please do. But until next time, folks, well done, and thanks for joining me, and class dismissed.